Welcome everyone. Thank you for attending today's presentation, How to Use Phoenix Reference Scaled Average Bioequivalence Templates. My name is Suzanne Minton and I'm a marketing specialist at Certara. If you experience technical difficulties in joining the WebEx, please dial 1-866-229-3239 if you're located in the United States or Canada. For all other countries, please dial 1-408-435-7088. To eliminate background noise, all phone lines will be muted for the duration of the event. Next slide, please. Please feel free to ask questions at any time during the event by clicking on the question mark icon on the floating toolbar located at the top of your screen. Please be sure to address your question to the default selection in the Q&A so that it goes to all panelists. We will address as many questions as possible during the Q&A session following today's presentation. This event will be recorded, and a link to a replay of today's webinar will be emailed within the next few days. Before we begin today's presentation, I would like to introduce our speaker, Anna Henry. Anna has been working for Certara since August of 2002. Since 2008, she's been the product manager of Phoenix desktop products, including Phoenix Wind on Lin, Phoenix Connect, Phoenix NLME, IVIBC Toolkit for Phoenix, and Phoenix Trial Designer. Anna has extensive experience in software demonstrations and training, as well as technical support for evaluations of software products. She has trained and provided support for Phoenix Wind on Lin, the Farsight Knowledge Base Suite, Autopilot, PKS Reporter, Trial Simulator, and PKPD methodology courses. Prior to joining Certara, Anna worked at Supercore Inc. as a clinical research manager and at Quintiles Inc. as a pharmacokineticist and biostatistician. She obtained her master's degree in the area of biomathematics from the North Carolina State University. I'll now turn it over to Anna to begin today's presentation.
Well, hello everyone. Welcome to this webinar where we will be demonstrating how to use the Phoenix Wind on Lin templates that have been developed to perform reference scale average bioequivalence. So what is reference scale average bioequivalence, or RSABE, which stands for reference scale average bioequivalence? First of all, to explain that bioequivalence is required for approval of generic drugs by regulatory authorities. In addition to generic drugs, a drug in development might also require to do some bioequivalence studies if the manufacturing site changes, for example, or the expedient in the formulation changes, etc. So a test drug should be bioequivalent to a reference drug for pharmacokinetic parameters such as AUC and Cmax. In traditional average bioequivalence, the 90% confidence interval for the geometric LS mean ratio for the test formulation over the reference formulation for AUC or Cmax must fall within 0.8 and 1.25, or 80 and 125 in percentages. So graphically, this point estimate, which is this ratio, and the 90% confidence interval should fall within this area of 80 to 125%. So in this case, these first three examples would be bioequivalent between a test and a reference, but other ones that fall, even if the point estimates fall within the range, but the confidence intervals are outside, it will not be considered bioequivalent. So reference scale bioequivalence methodology allows for wider acceptance limits. So you can imagine here that these lines, instead of being at 80 and 125, they're now wider, and therefore some drugs may now be considered bioequivalent. The extent to which these acceptance limits can be widened depends on the intra-subject variability for the reference product. And this methodology has supported several approvals of highly variable generic drugs by regulatory agencies. Okay, so why use reference scale average bioequivalence? Since the determination of the study sample size depends on within-subject variability for highly variable drugs and drug products, those that are defined as having a within-subject coefficient of variation greater than 30%, the sample size needs to show bioequivalence with the sample size that you need to show bioequivalence with traditional methods are very, very large. So just to give you an example, so if you had a highly variable drug product within subject variations of the reference product of, let's say, 40%, you would need a sample size of about 66 subjects to show bioequivalence using the standard BE. But if you were expanding limits to like 75 to 133, that sample size will be reduced to 28 for a partial replicate design, a 3-period, or 20 for a full replicate design of 4 periods. So in other words, this is a significant reduction on enrollment and cost if you use reference scale average bioequivalence for highly variable drugs. We also know that there exist some examples where highly variable drugs fail to show bioequivalence to itself when doing a standard average bioequivalence and using the standard sample sizes, which usually range around 24 subjects. Large sample sizes also means increasing expense and more subject risk. So reference scale average bioequivalence methodology is increasingly used to demonstrate bioequivalence with highly variable drugs and highly variable drug products. Why Phoenix templates? We have, you can perform reference scale average bioequivalence with Phoenix with non-lin 6.3 using reusable template projects and workflows. So currently, Phoenix only has a B module to do standard average bioequivalence, but reference scale average bioequivalence analysis can be accomplished via workflows. These workflows can be extensive, but the methodology is pretty fixed, so templates can really help you do this work and reuse the steps. These templates can be used by non-task programmers and by anyone who has a Phoenix with non-lin license. These projects require really minimum input from the user. There's about 3 or 4 steps for one of the templates and 5 steps for the other. And it can be used with any input data set for a replicate 3-period or a 4-period crossover design. These templates are available for free download at these links that we will be providing you. These templates are the result of a collaborative work with Helmut Schutz at BBAC Consulting, Simon Davis, Linda Huge, and myself from Certara. And this work was performed for a poster presented at AAPS this year. Okay, so I'm gonna talk a little bit about the regulations and using reference scale average bioequivalence. So the specifics on this methodology vary a little bit between agencies, so between the European Medicines Agency and the FDA, but both require the subject receive the reference product more than once to account for within-subject variability. So a 4-period full replicate design will have test, reference, or the next sequence, reference, test. And a 3-period partial replicate will have to have at least the reference twice. So you can accomplish this by having a test, reference, test, reference, and reference, test. So this would be a 3-period, and this would be a 4-period. For the European Medicines Agency, the methodology that can be employed is it's also referred as average bioequivalence of expanding limits. Requires that the reference product within subject variability is greater than 30%. And for the FDA, if the within subject standard deviation needs to be greater or equal than 0.294. Some specific requires for the EMA, those are taken directly from the guidance. The bioequivalence study must be of a replicate design that has been demonstrated that within subject variability for Cmax of the reference compound of the study is greater or equal than 30%. Sorry, greater than 30%. Okay? So just to demonstrate that, you need to have the reference given twice, as we explained in the 2 designs. You also, though, need to justify that the calculated intra-subject variability is not the result of outliers. So if there's some extra change, if there's outliers that are creating this variability, you need to remove them. So the extent of the widening is defined based upon the within subject variability. So those limits, the upper and lower limits, are now extended to have the exponential plus minus a constant 0.760 times the within subject standard deviation of the reference product. And in this case, for EMA, is for Cmax. Although there is also a limit to how far you can extend these limits, so you cannot just go on and on extending the limits. If you have a confidence of variation that is like greater than 50, then those limits are fixed, and then you can only use a 69.84 to 143.19. So the geometric mean ratio should lie within the conventional acceptance range of 80 to 125. Prior, we have seen that for the average, for the standard average bioequivalence, not just this ratio, but the confidence intervals need to lie within this range. Now, one of the requirements, if you do have highly variable drugs, is at least the ratio needs to lie within the range, and then the limits can be expanded. Okay, so you can have this possibility to widen the acceptance range for highly intra-subject variability being high, but it does not apply to AUC, so it only applies to Cmax. For EMA, AUC, it still needs to remain within the 80 to 125%, regardless of variability. That means the confidence intervals need to be within that range. Okay, some FDA-specific requirements. If the within-subject standard deviation of the reference product is less than 0.294, then you need to use the standard average bioequivalence methodology. This is the two one-sided test procedure that we've talked about. However, if this within-subject standard deviation of the reference product is greater or equal than 0.294, then you can use the reference to scale procedure to determine bioequivalence for individual PK parameters. So that's one of the differences too, is the FDA allows you to use this methodology also for AUC. So how is this determined? A 95% upper confidence bound determined by a chi-square test for this term right here must be less or equal than 0. And this term, the y here, is the mean log of the PK parameter for the test minus the mean log of the PK parameter for the reference, all this square minus this constant, theta. This is the regulatory constant indicated right here, okay, times the within-subject variance for the reference product. Now, it might be easier to see that these limits are actually being scaled by looking at a rewrite that we have right here on the bottom. So you see that now these are being scaled on both ends. And in addition, the point estimate, so that the ratio of the test to the reference, the geometric mean ratio, must fall within the 0.8 to 1.25. So when you download the templates that are available to you, you will get a zip file. And in this zip file, you will see what I have here. So you will see 4 files and 2 subfolders. These files right here are the templates that are developed. The one that I'm pointing at right now is for EMA, and the other one is for the FDA. In addition, there are some documents that explain how to use these templates, what we're going to go through today. And those are also included within the project. So when you open it in Phoenix, you will also in the document see these PDFs that you can view. But they're also included here in case you want to take a look at those before using the templates. We also have a subfolder called template copies. This simply has copies of those templates in case something happens to them and you need a clean copy. The other subfolder is an example of executed projects. So in that subfolder, there's several examples. So we have 4 projects that have been executed, 2 for FDA and 2 for EMA. They're using the same data. This one for FDA, it has the partial replicate, and it's already been executed. And this one has the full replicate, and the same for EMA. And also the data that has been used. This data was provided by the EMA in a revision 7 that they posted. In addition, we have some templates for the SAS, where those are confirmations. In all of these ones, you will have a workflow that shows you also some SAS that has been created via Phoenix Connect that just runs SAS to verify the results that we get in Phoenix. And you do not need to have SAS. You can just open those projects and you will see those results. Now, if you do have Phoenix Connect and SAS, then you can re-execute them. But without those licenses, you can still see those results and those projects without any problem. So hopefully this will help you if you want to start by just taking a look at things that have already been run and how it looks like. You can open these executed projects. Okay, so I'd like to start by showing you what is the result, what are you aiming at with these templates. In these templates, there's two final tabulations. The top one that I have here is for EMA, and the bottom one is the one that you get with the FDA template. So in this table, it uses a Whitnall-Lin table. It shows you this, the geometric LS mean ratio here, so that we also refer as the point estimate. It shows you the standard average bioequivalence, 90% confidence limits. It shows you here the coefficient of variation for the within subject of the reference product, and then, if applicable, the expanded limits that you could use. So in this case, it says, yes, I have highly variable drug, is greater than 30. I could use these expanded limits of 71 to 0.23 and 140 to 40. Although in this one example, you would see that we actually were meeting already the standard average bioequivalence limit, so you probably wouldn't need this methodology. For the FDA final tabulation, it has an assessment at the beginning. These changes depending on your data. So it would say things like you cannot use the reference scale average bioequivalence, or yes, you can use it, and then it tells you the criteria for bioequivalence is met or not. So in this part of the table is your average bioequivalence result, same as above. And then in this second part of the table, it just refers to the reference scale average bioequivalence, and it tells you right in the title what the criteria is to meet bioequivalence. So you need to have these within subject standard deviation of the reference product to be greater or equal than 0.294. In this case, that's correct. You need to have your point estimate between 0.8 and 1.25. This is also met. And then you need to have that 95% critical bound to be less or equal than 0. And in this case, it's also met. That's why it says in the assessment that this criteria has been met. And we'll show you that more when we show you the templates. So before I start the demonstration, I just wanted to talk a little bit about some considerations here, that we have used the test data that EMA has provided to do testing for both the full and partial replicate design. And we have also matched those results to the SAS that is provided by FDA in the progesterone guidance. And the EMA has also provided some SAS, and we have also matched those results to it. However, these templates have been tested by authors only and did not follow a formal testing and validation procedures for far-side software development. So this is something that we're providing to you or for you to use, but it's not really part of the software as far as you being able to call, for example, Certara support with the software questions. If you do have questions, you will contact us, and we're providing you that information later on. And also point out that these templates have been developed for Phoenix Whitnall-Lin version 6.3. So they really will not work with 6.2. And during the development of these templates, it was kind of interesting. We did run into some issues creating the template that you wouldn't really experience when you're using Phoenix Whitnall-Lin, but when you're using very, very large templates, you might run into some of these issues, and we have fixed them all for the next upcoming version, and all of them have workarounds that I will show you. So when I show you these templates, the steps that you need to do to set them up are very simple, and the steps that you need to do to execute them in a couple of cases just need to be very aware of how to do it. Those are workarounds about some defects that were found in 6.3. And just to tell you that in the near future, we plan to provide updated templates for the next release of Phoenix, that's version 6.4. This is coming up next year, so by mid-next year, we will have a new version of Phoenix Whitnall-Lin. And these templates will be updated, and we will provide you those links too. Okay, so with this, I will start the demonstration. I hope you can see it. I have Phoenix right now opened, and I have already loaded both of those templates, the EMA template and the FDA. And we will start with the EMA template. Once you load these, you will see that your workflow is red, because at this point, we really don't have any data. This is just a template. There's no data that I have loaded. Under this data folder, this is not real data. It just indicates the steps that you need to perform in order to use this template. And I'm just going to go through all of these, but prior to that, I wanted to show you that under Documents, There's the guideline, the EMA for bioequivalence, and we've attached it there as a PDF that if you just want to look at it for reference, you can find it there. And there's also instructions. So this is another PDF. If you click on it, you're not able to see it within Phoenix. You can just view an external viewer. So this will bring it up, and you'll be able to follow these instructions. Hopefully they're clear enough. They provide you some examples on how to fill some of the information that is needed. It walks you through some of the screens, and it provides you screenshots and talks a little bit about each of the steps, which is more or less what I'm going to be doing today. So that is included within your project. Okay, so the first thing that you need to do once you open this template is to bring your own data. So we're just going to go ahead and import some data set. We'll use these examples from EMA. These examples contain both the partial and replicate designs. So I'm just going to briefly show you this data. For the full replicate, you would see that subject 1, for example, has 4 periods, 1, 2, 3, 4 periods. It has formulation reference test, and then it has a sequence that has been labeled as BA, and other subjects have a different sequence, ABAB, which is then the opposite, TRTR. And there's, I think there's 78 subjects. And for the partial replicate, the data is similar. You have subject, period, sequence, formulation. This is data and log, the natural log of that data. You might or not have these. It's not necessary to run the templates. And in this case, we have subjects that have 3 periods, 1, 2, 3, and they have different sequences. So in this case, for subject 1 is RTR, and then if you see for subject 4 here, it's TRR, etc. So the first step is to upload your own data. Then it's also to save the project. It's very important that you save this project instead of saving it over the template. It won't allow you to do it anyway, but just to be in the safe, always when you're here, just go ahead and say File, Save As, Save Project As, and then save your project. We really recommend that you save your project often as you are running this analysis. So let's go through the steps that are outlined here. The first one, there's really not a step here. That's what is labeled 0. It's just an unformatted summary of instructions. So they're very similar to the PDF that I just showed you about the instructions. The second step, and this is the one that does require your input, is for you to enter the name of your reference. So in order to run these templates correctly, we need to know what your reference name is. So if it was something like drug A, you would go in here and you would type drug A, and you would need to type it exactly as needed. Here I'll also like to point out that in all the objects in these templates, we have added information about what each step or each object does. And you can go to the information tab. This is something that Phoenix has, and you can see here, you know, what are you expecting in these. For example, this is a file that you should review prior to running the project, and you need to enter the exact spelling for the reference formulation. And as we go through it, I'll show you some other information tabs. So here you type in what your reference name is. In that case, it's R, so we're going to leave that as the default. In the second step is to enter if your data is log transformed or not. So if you just had, for example, Cmax values, raw Cmax values, you would just leave these as an N. If you had the log of the Cmax values, you would then enter a Y right here. The third step is to enter your sequence. So if you would have 3 sequences for a partial design and only 2 for a full replicate design. So let's start, we will run the partial first. And if we go back to the partial replicate, you will see that the name of my sequences here is 1, 2, and 3. So in my enter sequence name, I would just put 1, 2, and then I need to put a 3. And this needs to be carefully reviewed. Just make sure that you're doing it correctly. And there's some examples of these, and you will see it in this demonstration too. So this is it, 1, 2, and 3 steps. And then we go to the workflow, and there's one last step, which is to map your data. So this is step number 4, and you need to take your data. We've decided we're going to do the partial replicate. We're going to drag it into this mappings panel. Now you need to have in your data set subject, period, sequence formulation, and a column of your data. Now these, they do not need to be called these names. You can actually map them. So if this is called ID, you would just put this dial button with the subject with ID. In our case, it has picked them up because they're called the same. We have said that the data was not log transformed, so I'm mapping it to data. If I had to put a Y on step number 2, then I wouldn't need to put to map log data type of column. Once you've done this, you're ready to execute these templates. However, to execute this one, the first one, which is prepare the data set for analysis, you need to do these step by step individually within this workflow. So this is one of those intricacies of running these workflows that we found in 6.3 that have been fixed in 6.4. But I just wanted to make sure that this is clear. If you try to run it from here, nothing will happen. If you don't see things happening, just go back here and then execute individually. So you execute this individually, and you can use the execution button on top, or you can also, to do it a little bit quicker, I just use the arrow down and then I press F7, and that just executes every step. And you see them flashing. So what I'm doing right now is I'm taking the data and I'm merging some of this information that you have entered. So we're merging the reference name, we're merging the numeric sequence, we're merging the transport flag, and then we have a data set that is ready for analysis, and we'll just take a look at it once it's executed. Okay, so as you can see, we have now this data that has been brought in, we have the flags that have been brought in, and we have taken the log of the data since it was not priorly log transformed. Okay, and just to tell you again, you know, if you're getting lost when you're doing some of these steps, you can always go to the information and find out what step, what's doing what. Okay, so now we're ready to run the analysis. Within the analysis workflow, we have 2 different analyses: the standard average bioequivalence and the average bioequivalence of expanding limits. And then you'll see that there's different sections within each. I'm going to go ahead and run them, and while it's running, I'll discuss some of it. So you'll see here, this is the standard average bioequivalence. We are running a bioequivalence wizard, and then we are tabulating those results, and then we're also doing other methods, and I'll talk about these. So I'll go ahead and execute this one. You can execute from the top level, so you can just highlight the standard average bioequivalence of the 2 little wheels, and you can just press execute. So the default for these templates is what we refer to as a model method A, and this is a model that only has fixed effects. Now there's other 2 models that have been discussed in the EMA guidance. One is called, the reference is method B and C. Method B has fixed and random effects, and method C is like a replicate type of design. And both of them are also available here for comparison, but the default currently that it's set up is for method A, and I'll show you how you could change that. Okay, so we've run the standard average bioequivalence. We can go to the table here, and you can see the results. So this just provides you a small table that shows you this bioequivalence table. Now this is for method A. Notice that other methods here are this method B and C, and you can also calculate within subject variability for method C. And you can look at the comparison results that will show you how these 3 methods differ. In this case, they're exact for A and B, but they're slightly different, you will see right here, for method C. If you wanted to use a different method, you would need to go here in this calculation step, the first step right there, and take one of these and just drag it into this window, and then you can select the output, your bioequivalence output from any of these methods to proceed to do the ABEL. calculation. So I hope that's clear. If not, we can talk a little bit more on the questions and answers. But at this point, I'm just going to continue with the default method, and you can have these expanded or not. We can go, we're doing two things here. We're doing calculations step and getting to final conclusions. These already been executed, so just ignore these for a second. And then we do an outlier analysis. So let's go ahead and run these. I'm going to execute these last steps, and then we'll take a look at these. While this is executed, I just wanted to mention a couple of things. As you know, there's two flavors of Phoenix. We have a 32-bit and a 64-bit, and both Phoenix, a 32 and 64, run these templates without any issues the same way. There's no differences. If you look at my Phoenix right here, this is Phoenix 64-bit. It's still Phoenix 6.3, version 6.3. This is just talking about the bits. And so we've done testing with 32 and 64 bits, and they run almost exactly the same as far as time is concerned, and they execute the same. So there's really no difference. Either of them would work. Okay, so we see that this is right now executing. When you see these locked right next to objects, is that they're either executing or they're being locked because they're inputs to whatever we're doing afterwards. So we're just going to give it a few seconds for this to finish, and then we'll review this final conclusion as well as some of the outlier analysis that is provided. Okay, so we can see here on the final conclusion, we have the table that I mentioned before. It shows you the ratio, the 90% confidence intervals for the standard bioequivalence. It shows you the variability, and then the extended limits if applicable. In this case, they're not applicable because you can see that your confidence of variation is not greater than 30%. And therefore, there's no scaling that should happen in this particular case. Also, for outlier analysis, we'll take a look at some of the output here. You can see there's a box plot that is available to you. And there's a QQ plot that is available also to determine if the student test residuals are log normally distributed. And if you do have some specifics about the data, you can go to the data wizard student test residuals and take a look at the student test residuals that have been calculated. And if there's a potential outlier, it would be flagged with these stars. That just means that this student test residual is greater than 2. Now, it means that you need to go back to your data and decide if this is an outlier or not. We're not making the decision for you in the template. And we're just pointing out that this could be an outlier and you need to look at it. Okay, so this is how the EMA template works. And just to dig in a little bit about what's going on here, just wanted to show you some of the steps and emphasize, for example, here we're calculating this confidence of variation. You can click on it, and you can see the steps that have been done. So this is quite large. You can review them. There's also some information here in the information tab. In this case, it's not that extensive, but it just goes through all the formulas that have been set up to create the results that we have demonstrated you. And there's several steps right here. You do not need to touch them again. The only one that you may want to do is to change the method of the average bioequivalence. So it's currently mapped to method A, but if you wanted a different method, you would then just drag it into the mappings panel for this one step. As an example, I'm going to go ahead and rerun this template and use the full replicate study now. And again, I'm sorry, I just need to emphasize that even though I'm not doing it from this demonstration, you do need to save these often. So, please do remember to save your project as you go. All right, so what I'd like to do now is just to rerun the same thing that we have done now that we know how to do it, but with different data sets. So we're going to be using the full replicate. Notice that there's a difference here in the sequence is labeled. It's not the 1, 2, 3 or 1, 2. It actually has some names here. My reference remains R, and in this case, maybe we're going to go ahead and use the log data. So in step number 1, I do not need to do anything because my reference is still R. In step number 2, I'm going to now change this flag to say yes. So it's yes, it's log transform. In step number 3, I need to enter my sequence. So to do this, we know this has not 3 sequences because it's a full replicate, only 2 sequences. So I remove this. It should be blank for full replicate. Then I type in here, what is the sequence that corresponds to TRTR? So in this case, it's AB, and for the RTRT is BA. But you are responsible to making sure that these matches. I'm just going to go ahead and expand these. So there's an explanation here. It does need to correspond to giving test, and reference for a full replicate. And for a partial replicate, it's with test, reference. So just make sure that these do match what your data is, what your data has. Okay, so we've set up this. Now we go to step number 4. We pull in the full replicate data instead of the partial. We make sure this is correct, and notice here that it's being mapped to data, not log transform. So I want to change that to be the log transform because that's what I indicated on step 2. And now we're ready to execute this. So we're going to do the same steps as we did before as emphasis. So we're just going to do this first one step by step. And I know you don't see it, but all I'm pressing F7, which is the same as the execute button. So we have merged the reference name, the numeric sequence, the transform flag, and then we have now a data that is ready for analysis. And we'll take a look at this data. It's a little bit different than the previous one. And you can see here that now we have the data corresponds to the same column because it was already log transformed, so no transformation has happened, and then we have this transform flag and formulation. Okay, so as last step, this will take a few minutes, but we'll go ahead and run this analysis, and I'm just going to run it from the top level, and you'll see that this starts flashing and showing you that this analysis is being run. While we wait for this, I'm going to start showing you some of the FDA templates. It's a little bit more complicated than the EMA, and the methodology is different, but it's set up with the same idea as I showed you for EMA. So you have under the data here, you have some steps to do, and notice there's one additional step for the FDA, and then under the workflow, you would need to map the data. So that idea remains. Okay, as this executes, the first step that I need to do in a template is to load the data. So we're going to go ahead and load the data while this is executing. And I've loaded the same data set, so we're now familiar with how this full replicate data set looks like and how the partial replicate looks like. They're the same data that we use for EMA. And I don't really want to continue with the FDA until this is completed, so you can, don't get confused of how the two templates are, because they're a little bit different. Okay, but since this is still going, we'll go ahead and just take a look at the first steps, which are very similar to what you did for EMA. The first one is just to enter the reference name. The second one is to tell us if you have a log transform data or not. The third one, which is different than EMA, is indicate if this is a full replicate or a partial replicate. So F is for full replicate and P is for partial replicate. So you would just need to change it here. The default is F. If you're doing the partial replicate, you will change this to a P. The fourth step is they enter the sequence name, so it's exactly the same, set up with the same idea. And these last two do not need to be modified. So please do not touch either A or B. Just leave them under the data folder as they are for the FDA template. Okay, so I'm now still running here. Going to give it just a few more seconds. Before we move on to the FDA template. Okay, so it's now done. Remember, we had run, first we ran it for partial, this was now for full, and we can look at the final conclusion here and see that for this data set, for the full replicate, we saw that, I think it's the same screenshot I showed you already in the PowerPoint, that we can use these extended limits because we have our coefficient of variation for the within subject of the reference greater than 30. However, really is not needed because you can see here that we're still within the standard limits. Okay, so this was for the EMA. I'm going to close it, and then we'll continue to talk about the FDA. We've already talked about the first 4 steps that you need to do, and at this point, really save your project. So I know I sound like a broken record, but it's very important that you save your project, especially with the FDA template, because it has a few intricacies on how to run it. And we're going to go ahead and do the full replicate in this case. So I'm just going to bring the data here, and we're going to have it mapped to not log transform. So let me just confirm that I have this corrected. This is not log transform. This is a full replicate. It has an F, and I do need to enter my sequence name. Now, don't be confused here because we're looking for TRTR, and if we look at the data for the full replicate, you will see that TRTR is this, ABAB, okay, TRTR. So when we go here, in the sequence, ABAB. And I'm emphasizing this because once I looked at this, I noticed that one of my examples is opposite. So do not get confused by these examples. You do need to just look at what matches what's expecting. So in this case, it's ABAB and BABA. We have the data that is here mapped. This is not log transform. So we go ahead and run. Now, similar to EMA, all these steps need to be run one by one. They need to be executed one at a time. So you need to wait until they are finished the execution and then click on the next one and continue. As you can see, there's already a lot more steps in this one because the preparation of our data for FDA is a little bit more complicated. So we're not just merging the information that you gave us, but we're determining the order of the formulations, and I'll show you that when we get to that step. So in this upcoming step, what you want to know is which one is the reference given first, so we call that R1, which one is the one given second, we call it R2, which one is the test given first, T1, and which one is the one given second, T2. So it just determines the order the formulations were given for each subject. You will see right here that now this indicates this is R1, T1, R2, etc. And we'll continue. The next step of appending worksheets, this is nothing you need to be concerned about. This is something that I needed to do for this template to work correctly. The next step that we just did here was to take the log of the columns, and now I'm getting a data set that is ready for analysis, but it has subjects that received all the R periods. So if there was somebody that for some reason in the 4 periods dropped out and did not receive a second reference, they're removed from this data set. And in the second one is all the subjects, they have to have received all the periods. So notice that if we look at this data now, we only have 1 entry per subject, and this has been pivoted. So now this is R1, R2, T1, T2 going across. To run the average bioequivalence, you can just highlight that workflow and execute it. And you will see that this will provide you also a table. And the next step is the one that does require a little bit more attention. So this is something I really wanted to show you to make sure that nobody's confused on that. And I'm just going to wait until this finishes so I can show you clearly. And we see that as they turn black, these are the steps that are happening. It's calculating the coefficient of variation here for the within subject reference, then merging and getting you some of the average. So these are, again, just the average BE, but it already has the calculation of the coefficient of variation. So this is the one that you will need for later on. Okay, so here, to run the reference scale average bioequivalence, there is, in the data wizard, there's a defect in 6.3 that, depending on how many steps there are before this happens, it might run into some trouble. So you might execute and it might not move at all, nothing would happen. So before running these, please do save the project. And this has been fixed in 6.4, but I just wanted to show you how to run these. How to run these is, you need to go highlight these, the data wizard, go to the setup tab and highlight the last step. But every time you highlight it, it needs to flash. It needs to do this. Okay, so it's doing something even though you have not clicked the execute button. That's actually a good sign. If you do not see when you highlight it, if nothing happens, then you need to go to a previous step and then back to the last step. So make sure that every time that you are ready to run this data wizard, that you highlight the last step and make sure that this is happening. So you're not doing anything, it's just flashing, okay? And this is actually a good sign if it's flashing, and then we'll be able to execute it afterwards. All right, so I'm going to wait until these finish flashing. And in the meantime, I also wanted to tell you that the location where you can download these templates has been pasted in the, I believe it's the Q&A. Linda has probably pasted this, and you can have it electronically. You can just copy paste to find those templates for you to download. Okay, so now this is ready. You notice this took like a minute. This should happen once this is, make sure the last step is highlighted, you're here, and then click execute. If you do it exactly this way, this is going to work. If not, you may run into a situation where it's saying that it's executing, but it really is not doing anything. So if you wait, I don't know, I would say more than 2 minutes, then you probably have gotten yourself in a situation that is not ideal. If you click on these, soon we will see that here on the side, these are going to start flashing like that. So if that's the case, that's fine. That means that this is going and it's not a problem. But if you do not see this flashing and you've waited already a few minutes, you might have found that defect that is in 6.3. So just make sure that you actually run it the way that I just indicated. And then for the final conclusion, you need to run these individually too. And you don't need these really, it's just to provide a really nice table because you are merging all the results from the standard average bioequivalence and the RSABE results, so you can look at them side by side. I think I mentioned that this is due to some of the data links that are within this sub workflow that you need to run one by one, and it has been fixed in the next version of Phoenix 2. Okay, so we have now our table. It's the same table I showed you in the PowerPoint, indicates that yes, the criteria has been met for this full replicate because it meets this criteria indicated on the top, and if you need the average bioequivalence, it's here. All right, so due to time, I am not going to do this for the partial. And I'll probably right now will conclude my presentation and have Linda Huge assist me with the Q&A, because we want to make sure that we answer some of your questions.
Hi, Anna. The first question we have is, can I use these templates if I have several PK parameters in a column, such as AUC and Cmax?
Oh, yes, excellent question. Unfortunately not, because the way that we are mapping the data, if you take a look at this, it just only has one column for data. So these data right here, this column should be APK parameter. So these should be Cmax. If you had AUC, then you would need another one of AUC, and then you would need to run the template again for AUC. So excellent question, but no, just one parameter in a column.
Okay. Next, would you be able to demo now for the EMA how to use method B or method C instead of method A?
Sure. Okay, let's do that. So we're going back to the EMA, and let's make sure that I know which one we have right here. We have mapped. We are doing a full replicate, and you now would like to use, let's say, method C, okay? So to use method C, it's already been executed within the standard average bioequivalence sub-workflow. What I need to do is expand these calculation steps and highlight this. This is a data link. If you go to the information, it would tell you, it brings into this workflow the results from ABE. And there's a typo here, sorry about that. This is method A is actually the default on the template. Okay, so if you want method C, you're going to take it from here and just map it. So you're dropping it, and then it's showing you, what would you like to use. You need to pick the average bioequivalence. So I tried to help here by putting the name right in there. So this is the worksheet that you're looking for, average bioequivalence. So select this one, click OK. It should find the same exact names because the other method is the same object. So it's already all mapped for you, as you can see. So it's already finding all the mappings for you. And from there on, then you just need to execute. So we can just go ahead and execute this whole thing using the different method. I'll just do Okay, go ahead. Let's do another question. Well,
and are the instructions for using method C like that included in the PDF file?
Yes, good question. Yes, they are, and I hope that we made it clearly enough. There's arrows indicating where things should be mapped, just as I just did for you.
Anna, the next question is, can these templates be modified?
Yes, of course. This is not a module within Phoenix with Lynn. They're just templates that you could have created the same as we did. They can be modified, but these particular templates are read-only, so you can modify them as you go, but when you go to save it, you will need to save them as a different project name. So that would be the only difference. But yes, they're modifiable as you go. Just make sure to save it under a different project name.
Okay, and the next question is, does Farsight offer template development services?
Yes, actually we do. We do offer template development services. So if you're interested in templates that are different, that doing these methodologies, just other type of work that needs to be repetitive, we do offer those, and you can just contact sales@certara.com. And as we speak about that, I'll bring in some of the contact information. So for here, if you require more information regarding these presentations, you can email me or you can email Linda. And also to point out, there's other resources. There's a video in YouTube about how to create templates in Phoenix, and I put the link right there. If you're interested in seeing it, it's not specific for reference to scale average bioequivalence templates. It's just general on templates in Phoenix. Could be a good resource too.
Okay, then we have a question. A user has version 6.2, so they'd like to know what they should do about that.
Okay, so 6.2 is different than 6.3 in the sense that you don't have those data wizards. So if I go back here, you will notice there's a lot of data wizards that have been included in these workflows. So the steps within that data wizard you can do individually in 6.2, but these templates will not work. So my only recommendation at this time, I don't think we, Linda and I right now will be creating templates for 6.2. We'll only go forward with 6.4. But Helmut Schutz do have templates that were created for 6.2 for the reference scale average bioequivalence. They're probably not as clear as some of the steps here, but some of the basics are there, and you can download it from his website. And I don't know, Linda, if you have anything else to add to that.
Right. Well, there's also the upgrade path, right? The users can contact TRTR about getting an upgrade to 6.3.
That's correct. Of course, if you're willing to upgrade, and just with your current license, if you pay the maintenance fee, you can upgrade to 6.3 for free.
Okay. And the last question that we have, a user is asking about a template for Tmax. I'm not completely sure about their question. They may be asking about using a non-parametric method, but what they're asking for is a template that will work with Tmax.
That probably can be developed, but that's not something we have currently developed. And I don't know, maybe we can think about it for next time around. Now, no promises, sorry.
Okay. So that concludes our questions.
Okay, excellent. Well, thank you very much, everybody, for attending. Once you log off, there's going to be a survey, and we will highly appreciate if you could answer those 3 questions that will be posted to you. Thank you very much.